Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society. Hi, I'm your host, Dawn Cornegas, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's director and chairman of the double secret selection committee that selects all the guests who appear on STEM Talk.
Hello, Don. Great to be here.
So our guest today is Dr. Elaine Choung-Hee Lee, an associate professor in the Department of Kinesiology at the University of Connecticut, and much of her research focuses on understanding the mechanisms of resilience and also studying ways to help humans improve their stress resistance, adaptation, and also health span.
We've enjoyed getting to know Elaine over the past few years. Her research is not only focused on understanding how the biology works, but also what people and researchers can do to manipulate our biology to optimize health and performance, as well as preventing disease.
And at her UConn Research Center, it's called the E.C. Lee Laboratory, she and her colleagues use a variety of approaches to ask questions about what makes high-performing athletes and also warfighters so elite. So in today's interview, you're going to hear about an early passion for Marvel Comics and how superheroes helped nudge Elaine into a science career. You're also going to learn about some of her lab's projects that range from improving warfighter resilience to studying the effects of exercise and supplementation on our immune function. But before Ken and I get to our interview with Elaine, we have some housekeeping to take care of. First, we really appreciate all of you who have subscribed to STEM Talk, and we're especially appreciative of all the wonderful 5-star reviews. As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes, Google, Stitcher, and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Today, our winning review was posted by someone who goes by the moniker Liz E. The review is titled STEM Smart. It reads, "I feel so smart when I listen to STEM Talk. The topics open a whole new world for me, one that I can share with my elementary school students. I especially love when I can follow along and understand certain topics because of the sciencey background I'm building with each episode. Thank you for the variety of topics and guests you bring on your show. Signed, sincerely, Ms. Liz."
Well, thank you so much, Ms. Liz, and thank you to all of our other STEM Talk listeners who have helped STEM Talk become such a great success.
Okay, and now on to our interview with Elaine Choung-Hee Lee.
STEM Talk. STEM Talk. STEM Talk.
STEM Talk.
Hi, welcome to STEM Talk. This is your host, Dawn Cornegas, and joining us today is Elaine Choung-Hee Lee. Elaine, welcome to the podcast.
Thank you for having me.
And also joining us is Ken Ford.
Hello, Don, and hello, Elaine.
So, Elaine, I understand that you were obsessed with Marvel Comics and superheroes. So how old were you when this obsession took hold of you?
Yes, I'm really interested in heroes in general. I would say when I was younger, I had a real big obsession with Greek and Roman mythology. So even when I was about, I would say 7 or 8 years old, I knew at that time all the Greek gods, even the minor ones, their powers, the interrelationships among them. But I don't think I really got into Marvel specifically until the big companies started putting out cinematic, huge Marvel movies in the 2000s. I think Iron Man was the first one. So I've always been really interested in heroes, but I think Marvel has been a really recent interest of mine with, everything that Disney is doing.
Yeah, I agree. So now we have to ask, who's your absolute favorite Marvel comic hero?
So I have a few that I really kind of follow and am interested in, but I would say my favorite is Captain America.
So what's so special about him?
I think Captain America right now, as for me as a scientist, and then also kind of what I am inspired by is that he is among the heroes the most real to me. He's a military veteran and he doesn't have necessarily what I would consider like unattainable, like magical powers. He's super strong, super fast, incredibly resilient. And I think that to me, there are human versions of that in our elite athletes. And so I think there's sort of like a tangibility to his heroism. And then the other aspect that I really appreciate about that character is all aspects of kind of the tragic nature of how alone he can be, and yet he's still really noble and he always does the right thing. And so there's so much about that character and person that I find really interesting and intriguing.
Yeah, I totally agree with you. So, what's your all-time favorite Marvel movie?
Oh gosh, it's gotta be, Winter Soldier is probably, one of my favorites, and then, of course, the big epic, Civil War and the last one that we just had. They seem to get longer and longer, right? So, the big long ones is what I would say.
So, shifting just a little bit, when did you first know that you were interested in science? When did the bug sort of bite you?
I think, the answer to that question is a little bit tricky for me because I don't think it happened acutely. As a student, I would say in 5th grade, our school in our local town in Connecticut had a program called Talented and Gifted TAG, and many of the schools here in Connecticut have that. And that was when we were first really introduced to science outside of the classroom. So we did, kind of projects in environment and whatnot. And so in that program, I think I really became interested, and it was just a journey that like I started to do more of what I liked, and the more I did, the more I enjoyed it. So I would say that program probably initiated the process for me to the exposure, and I think I just have enjoyed the journey that I'm on. So it's kind of interesting to think about from my perspective. If something else had kind of taken hold at that age, would I have be in a totally different place right now? But this is the journey that I've been in, and I really enjoy it.
Yeah, that's awesome. So, growing up, you had a couple of other obsessions, such as rowing and running, and you even became a rower at the University of Connecticut. So, what drew you to running and rowing?
Running is something I started in middle school, and I'll be totally frank with you. I did this sport because our family was very poor and we could not afford to do any other sport.
Mm.
So at that time, all you needed was a pair of decent sneakers and you could try out for the track team, and that's how it started. I found that I was a natural runner, and later on in my running career, kind of in high school, I ended up talking to my parents and discovered that my dad was a very successful long distance runner in South Korea. And so I guess it was built in, even though I didn't know it. And so that's what started me on running. And same thing, I had an opportunity, I started, I gained experience, and I really enjoy it. It suits my nature, which I think rowing does also, where I really enjoy kind of counting miles and pushing, how fast can I go and how hard can I go and all the data that I get with it. I think running and rowing are so consistent in the nature of the way you do the sport physically that suits what I enjoy and what I, get most pleasure out of it. Rowing is an interesting story as well. So I went to the University of Connecticut and I was not recruited to be on the track team, but in my freshman year, I was in the process of walking on to the track team. And I saw a flyer advertising for interested athletes to join the UConn women's rowing team, which was just at that time, I think about 5 to 7 years as a Division I program. And I was really intrigued. I had never been on the water or in a boat in my entire life. And the first kind of recruiting visit, they took us to the lake, the boathouse, and they put us in boats. They spread all these new recruits out among the boats, and I was hooked. I had never been so close to water or been on the water, and I was like, this is incredible. So
Mm.
after probably at that point having run for like 15 years of my life, and the translation, I think, is really good to rowing for the distance that I ran. So I was an 800-meter, 400-meter runner. I just found it was a natural fit, and I really, really enjoyed that experience.
What was your rowing experience like at Connecticut? Did you enjoy that a lot? And, what events were you in?
I enjoyed it immensely. I ended up coaching during grad school, coaching a little bit as faculty as well. I still stay really involved with the team, and I have done research on rowers. So I would say it's become part of who I am. At UConn, I think it was a really good chance for me because it wasn't a really established, entrenched rowing program. So we're not talking about, an Ivy League rowing program that goes to Henley Regatta every year. So there was a chance for someone like me who had never rowed at age 18 to join. So as a rower, I started off as more of a lightweight rower and in the JV boats. And then by senior year, I was in the varsity 8. I rowed mostly in eights and in the bow pair because I am a little bit shorter. So I'm 5'7", which is pretty short for a rower. But I think the technicality of my rowing and kind of my fitness and endurance contributed well to that role in the boat. And yeah, I think I just learned a lot also about sport and science, even outside of academics. So I'll give you one example is at that time, and probably still now to this day, track programs and high school athletics programs don't put a ton of emphasis on strength and conditioning or resistance training and even diet. It's really a lot of times coaches that are doing their best at the high school level. If you're not in an elite program, it's really kind of anecdotal experience and the coach's experience that drive the success of the teams. At UConn, it's a Division I program that's very internationally well renowned. And so I think I got a totally new experience in terms of lifting. We were practice on the water, 5 to 6 days a week, lifting really heavy 3 days a week, really good counseling and precise counseling on diet and, pre and post exercise consumption for recovery. And that really, I think if I had stayed on the track team, would have made a huge impact on me as an 800-meter runner. So I just learned a lot about kind of things that I didn't understand in sports science in that program. So rowing really, I think, changed my life.
You're the second rower that we've had on STEM Talk. Don, do you remember the interview with Brianna Stubbs? She was also a rower.
Absolutely, yeah, and a great friend and colleague of ours as well. So, Elaine, in 2002, you graduated from UConn with a bachelor's in nutritional sciences. When you set foot on campus, did you know that was what you wanted to major in?
No, it wasn't. So I feel like that's going to be the theme of the answers to many questions today. And that's okay, is what I always tell my students and my mentees. So I started out as a molecular and cell biology major. I think that comes from, my parents were first generation immigrants. We grew up with a really hard life, kind of a lot of struggle. And to them, their experience was that medical school and the track to become a physician was kind of this dream way out the change for the next generation. So I started out as pre-med with kind of that pressure of that generation on me. And I found quickly that I didn't think I was enjoying it. I liked science and I really enjoyed the academic work, but I found myself kind of wondering what more was out there every time I would sign up for classes and would see these other class titles in the course catalog. I was really curious and kind of like realizing that there's more out there. And I took a nutrition class and it was astounding to me how easy it was because I was so interested in it. I think I read the textbook within a week and a half because I was just like floored that there was a science about food. And I switched majors. And that was a really hard fight for me, generationally, culturally, because, my family and my culture at that time didn't really understand, like, what can you do with studying food and science and nutrition? And so that's kind of how I ended up in that major. But it was a really big decision for me. And it basically set me on this path to where I am today.
So you've written that your passion comes from your innate curiosity for everything. So you go on to say that the focus of your research and career grew from your own experiences as a runner and a rower and also a coach. Kind of touched on this a little bit, but can you expand on that a little bit for us?
Yeah, so I think when you look at the work that I've done and my publications and the projects ongoing in the lab, it really appears quite diverse and sometimes disconnected. But the main thing that I would say, I tell folks when I'm trying to talk to them about what we do is that, as an athlete and a coach, I have always been really curious about how some people are so tough and resilient and able to overcome pain and excel physically. And then as an athlete, trying to strive for that and how to push my own limits. And then as a coach, how to inspire that and motivate that and put into practice things that will help other people push their limits and reach more than they think is their maximum potential. That experience and that the drive about understanding that process more is really at the root of every single project that I do. So, for example, one project that we pursued last year that's totally unique was a study of the deterioration of running shoes over 100, 200, 300, 400, and 500 miles. And that came from my curiosity about, okay, well, if we have this incredible runner at the elite level, what's that extra little bit that we can support them with that may be related to gear and how often you change your shoes that could really prevent injury or help them feel better when they're training? And can we make some really good guidelines around that? And we found some really intriguing data. We found that among elite cross country and track and ultra endurance runners at about 200 miles very consistently, there was a change, not overtly, in the way that the athletes were impacting with force and how fast they were running, but they were making compensations to their form that we could detect with video. And so we have this working hypothesis now that, and I'm sure corporate and industry partners would be really interested in kind of taking this knowledge to consumers, is that we think that athletes should be on a subscription model where every 200 miles they should be switching out their shoes. And for some athletes, at least the ones that we tested, that's every 2 weeks. And that certainly is not the case. So I think that whole curiosity and experience as a rower and a coach brings me to questions like that. And it's really the root of every project that we pursue.
It's really interesting. And so it seems obvious that your early experiences in genetics and nutritional sciences as well have also played a major role in your career. So what were some of those early experiences?
Yeah, so I think one of the things as a coach and a rower that we did a lot of work on back in, 1999 to 2002, and then extending a little bit into my graduate time before 2010, was trying to understand customization of diet. And this was before we really started to hear personalized medicine, precision medicine, genomic medicine in the literature and in the media after 2008. And there was an understanding and appreciation among the coaches and staff at UConn and the nutritionist here, Dr. Nancy Rodriguez, who was a mentor of mine, that this general recommendations for timing, amounts, composition of foods and recovery were not really able to address issues with every single athlete, especially among females. And so I think that experience and then my current study at that time in nutrition and understanding of genetics and diversity in response to diet and response to different stressors really came together in those moments. And that was where I saw an opportunity, maybe 10, 20 years down the road to like basically where I am right now to say, okay, obviously one plan doesn't work. How do we customize that and make that more precise to help people reach their maximum, help them become more resilient and push their limits?
Hmm. So it's really interesting. So, just listening to you and just kind of going through the work that you've done up to this point, Elaine, it appears that you're not only interested in how biology works, but what people and researchers can do to manipulate it in a way that could result in functional change for broader populations. Do you think that's fair to say?
Yes, that's 100%. And I think that is something that I'm really happy about and proud of in our lab is that we are really curious about mechanistic science. But I think on every project, and I'm thinking back now and I'm kind of verifying in my head that we always have a person who kind of keeps us in line and says, well, what's the application? What's the next step from that we can take to the field? For example, if we have an idea about biomechanics, kinetics, kinematics related to shoe deterioration, okay, well, how can we detect that faster? What's the biomarker that can detect that if we take a sample of blood? And so how do we use that information to help the end user, the athlete, or the coach? And that's present in every study, even in the C. elegans studies that we do quite a lot of work right now with Caenorhabditis elegans. And even though it's a worm and it's microscopic and it doesn't seem like it relates to exercise, a lot of the dosing and supplementation studies that we have in C. elegans, we're taking directly to support the human studies.
You stayed at the University of Connecticut to get a master's degree and a doctorate, and you studied kinesiology. Could you tell us why you picked that? I mean, I can sort of hear from what you've said already why that might be of interest, but I think you might have more to say about that.
Yes, and again, that's not an intentional decision, but it was me taking a moment to walk through an open door So at that point in my career, I was a nutritional sciences major in my undergraduate, and I was still thinking pre-med because I think I was kind of torn, like not giving up this idea of a dream or something that, my parents and my family and people around me had kind of said, this is the way to go to the next stage. And I didn't get into med school. And so that was a really hard time for me. And I remember that I talked to a really great mentor of mine, Ellen Darrow, who was at that time head of the dietetics program. And she said to me, I think I have a friend who can help you. And that was Carl Marish, who became my graduate advisor. So he was my master's and PhD advisor. And I basically went to him the week before the semester started, told him my story. I knew very little of the actual science of exercise because, again, I had only been studying molecular and cell biology, genetics, and nutrition. And he accepted me. And the day before classes started, I had a GA and he started me in the program. I think he saw something in me in terms of my passion and my interest in who I was. And I took one class in exercise physiology 1. And same thing as when I took the nutrition class in my sophomore year, I was hooked. I couldn't believe that this was a thing that people studied every day of their lives. And it wasn't work, but it's work. And so I stayed for my master's and doctorate. At that time, that was incredibly serendipitous because even though this was a quote unquote backup option for me, I was very grateful to Carl for giving me that chance because at that time, that program was #1 doctoral program in kinesiology in the nation. So I really think kind of fate and destiny guided me to a path that was suited to me and who I am and what I'm curious about. And so I stayed for my master's and PhD in that program.
So, Elaine, after receiving your PhD, you headed off for a postdoctoral fellowship at the Mount Desert Island Biological Laboratory in Salisbury Cove, Maine. How did that opportunity come about?
So I actually had 2 postdocs. And when I finished my dissertation, which was a first-time biomarker look at some novel transcription factors that respond to dehydration, I came up with that idea. I looked into the cell culture literature to find how that might be happening in exercise and dehydration. So I did that project. And then at that point, I realized that if I wanted to create a program off of this, that I would need training in animal models. And I just hadn't done that. I've only done that in small internships in different ways at UConn. And I really wanted a focused training. So I went to Yale first, Yale School of Medicine, to do a postdoc. And in that postdoc, I studied ways in which stress and markers of stress and genetic control of stress in lymphocytes and immune cells could regulate how disease progression occurred. And so I did a postdoc in mouse work at Yale for about a year. And then to complement the mammalian piece, I also was thinking about, the first 5 years of my academic career. And I realized very early on in my postdoc at Yale that mouse work is extremely expensive. I did not know that. And so within a few months, I was thinking, hey, I might need another animal model so that I can do human work, mouse work, but have something that generates data faster. And so I applied to all these other labs for a second postdoc. And I came across Kevin Strange's lab at Mount Desert Island Biological Laboratory. He had been at Vanderbilt and had gone up to MDIBL to start this lab or expand this lab from a summer research institute into a full-time year-round world-class research institute. And that lab, MDIBL, has historical ties to Yale and kidney physiology research. So that was the transition. I went from UConn to Yale and then to MDIBL, and I basically went down the evolutionary tree from humans to mice to C. elegans in that process, always keeping kind of osmotic stress and dehydration stress physiology as kind of the theme through that training period.
So, Elaine, during that time, you and Dr. Kevin Strange, who's now a professor in the Department of Anesthesiology at Vanderbilt Medical Center, you two co-wrote a paper that appeared in the Journal of Federation of American Societies for Experimental Biology. And the paper was titled, this is a quote, Osmosensitive Gene Expression in C. elegans is regulated by conserved signaling mechanisms that control protein translation initiation. So in 2012, the Cellular and Molecular Physiology section of the American Physiological Society actually selected this paper as one of 6 finalists for its annual Research Recognition Award. So first, you must have been very proud of that recognition. And second, what was it about this paper that attracted so much attention? And
So
third, congratulations.
uh, thank you. Yeah, so this paper was foundational for how I kind of view science about stress right now. At that time, about 2008, 2009, 2010, there was this big advance in high throughput sequencing that really facilitated personalized medicine, precision medicine, genomic medicine, and at that time, a technique called RNA-seq, which allows us to measure the expression of thousands of genes at once. So I basically jumped on the bandwagon with Kevin at that time. I learned the techniques, I got trained, I went down to the Broad Institute during my postdoc, and this project started at first in an attempt to understand how C. elegans and human cells, by translation, respond to dehydration. So when the cell shrinks, the volume decreases. There's so much damage that happens in that process because the environment is not ideal. Charged regions of proteins become exposed, and there's damage that kind of begets more damage, and so on. And we wanted to understand when the responsive genes that help protect against that or repair damage turn on and how they're controlled during that process. But when we started to do these RNA-seq experiments, what we realized was the big groups of genes that were changing were not necessarily related to what you might think about. So they weren't necessarily immediately always enzymes that make osmolytes, which support the environment to prevent volume loss. They weren't always damage repair or chaperones like we would anticipate and how I thought would occur based on some of the human exercise work that we had done. And instead, what we were finding was that immediately at the onset of stress, environmental stress exposure or dehydration, the cell machinery shuts down, it slows down all protein synthesis. And that regulation is happening at the very first step of translation initiation before mRNA is translated into protein. And so we developed a new working hypothesis that supported an NIH grant, that supported my teaching T32 fellowship as a postdoc, where we think that, and this was confirmed by all this historical plant physiology literature in this area. So we were like, okay, this is not that new of an idea. But we had the hypothesis that at the onset of dehydration or heat or any other stress, whatever the cell's sensing mechanism is, and that still remains little understood in many contexts, that part of the major shift is to shut down protein synthesis to a degree, not completely shut it off, but slow it down so that the cell effort and energy partitioning is not focused on reproduction and growth as it should be under normal healthy conditions, but instead shifted to protection, repair, and damage recovery. And that is the key to that paper. And we were able to demonstrate that the translation initiation complex at a particular subunit, which is called EIF2, is inhibited by phosphorylation, and that the enzymes that are signaled to by that switch, that control, are also shut down. And we started to manipulate all these components of that complex. And what we were getting were worms that were incredibly resilient to salt stress. And not only that, but also chemical stress and other types of stressors. And it was so interesting because they don't all overlap. So we didn't find that, for example, worms protected by this mechanism against dehydration were also protected to heat stress in the same way, which I would have thought they were. And so that paper and that poster and the work that came from that in the subsequent years really focused around this concept that stress sensing and stress regulation is really predicated on this ability to shut down normal function and upregulate stress function and then come back to normal again in recovery.
Really interesting. In addition to the recognition that this paper received, you were also selected as the Mount Desert Island Biological Laboratory's Outstanding Mentor of the Year in 2012. So you must enjoy teaching and mentoring perhaps almost as much as doing the research itself. Is that true?
I do. And that's, I would say, the two most important things in my work and career and my goals for what I want to achieve are not just great science that contributes to people's understanding and some tools that we can use to help people be more healthy and more resilient and push their limits, but also allowing trainees and young people, especially those who don't have great mentoring or access, to see what they can do and empower them. And that comes from my life and my background. Like I said, I grew up with a really hard life in very poor economic circumstances. And so it really drives me to try and find students who are under-mentored or, lack confidence, and to help them see what they can do. And it's really astounding, I think amazing to students who are freshmen who come from high school programs, sometimes from disadvantaged or under-mentored backgrounds, and they're doing, PCR or some library preparation for RNA-seq, and they're seeing that it's following a recipe and the ideas are the most important thing. And so I feel really great when they feel like very proud to do that work. And so that's where the mentoring piece comes from. And then I think in my life, nothing has been given to me. And so my parents have always instilled in me and my siblings that you just have to always do your best in everything you do. And a lot of these awards come from that mentality where it's my job to take what I've been given and push those limits and try and open up doors for myself. So I have a funny story actually about this Day of Pride scholarship from UConn. Every year, only 10 to 15 students out of the entire university undergraduate. So each class is about 5,500 to 6,000 students. Only 10 to 15 from economically disadvantaged or under-supported backgrounds are chosen for this Day of Pride Scholar. And the goal is for UConn to support rising stars or talented students who basically have been given no tools. So that was like my life story at that point. And I remember that I had gotten into some really reputable colleges at that time, and some with some support and scholarship and loan packages, but our family was so poor that we couldn't even consider getting a student loan. That's how poor some students are, and we see that today too. And so it was devastating to me because I had these dream schools that I had gotten into, but I just couldn't afford to think about going to Boston or New York City or whatever. And so this Day of Pride Scholar letter was actually really discouraging to me. I was angry that I had to go to UConn, and it turned into like one of the best things. So I do support that program as an alumni now, and I take a lot of those students into our lab and try and tell them like, okay, if you didn't get to go into X and X and X, you're not in a bad situation. But that experience has also kind of put a perspective on the way that I mentor and teach in science.
So, Elaine, your research over the years has focused on understanding the mechanisms of stress resiliency, as you've been talking about, and ways to improve stress resistance, adaptation, and also health span. So, how did you become interested in this angle of research?
So I think the natural progression towards stress resilience has come from kind of what we've chatted about today in my time as an athlete and coach. But the relationship to health span and aging came, I think, because of that work with Kevin Strange and the pathways that we identified. So in 2012, when we started doing this work in genetic regulation and pathways that control how you respond to stress and how well and quickly you recover, those genes at that time in those experiments not only conferred stress resistance in naive animals, so animals who had never experienced the stress, we would genetically manipulate those components of the pathway and find that they were more resilient to dehydration or heat or hypoxia or chemical stress like juglone, which induces oxidative stress in the cell. But at the same time, the animals were living longer. And so when we dug into that literature, which was new to me at that time, I was finding, much like some of your previous guests on IHMC STEM Talk, that these pathways are really interconnected. And now it makes sense to me that if you are studying ways that animals can become more resilient and tolerate stress, that you may be also manipulating the pathways that control how healthy they are as they age. And so that's the tie-in, and that's why we've also kind of basically dipped our toes into this field of aging and longevity and health span research.
So, Elaine, you spend a lot of time investigating genetic and molecular mechanisms of whole body and cellular stress in C. elegans in humans and how it's caused by such things as heat or exercise or osmotic stress, again, as we've kind of been talking about. Can you talk about C. elegans and why they're so useful in your research?
So Cyanurhabditis elegans, or C. elegans, is a microscopic worm. It's a soil nematode that's also found in rotting fruit. It's a really interesting trivia fact that there were some studies done in the 2000s that identified that just random survey of big groups of people found that like 30% of them have antibodies to C. elegans, meaning that you've been exposed to them or consumed them at some point. So they're very common. And what is so amazing about C. elegans is that they're really genetically tractable. So their development from egg to adult is only about 3 days, and you can culture them in Petri plates in the thousands. So you can have an N size of 1,000 very easily in 3 days. They're hermaphrodites, so they are essentially clones of each other, but they have enough variability that I think it does translate to kind of the generation to generation variability you see among mammals and more complex organisms. And you can do anything with them. So they're see-through, so you can do some really cutting-edge imaging with high-powered microscopy. You can stain features of the cells so that you can do fluorescence microscopy. You can track one thing that's always been so amazing to me is their reproductive tract is linear. So you can actually see, let's say, 8 to 10 eggs in a row, and the first egg will be the least developed, and the last one right before it's laid will be the most developed. So there are people who've done a lot of work in reproductive development in C. elegans. The genetics genomics is complicated in that there are about 25 to 30,000 genes/isoforms, and there is about 40% homology to human genes. So in my area, in stress and dehydration and heat stress, many of the genes that C. elegans use to survive that stress are similar to human cell genes. So they're either exactly the same, like the enzymes that produce glycerol in the cell, for example, or they're very similar. And so you can also induce mutations in C. elegans by exposure to like ultraviolet light or radiation, and then track which worms respond and pick out worms that you're interested in studying. So you can do forward genetics, and then you can knock out genes very easily by feeding worms bacteria that produce double-stranded RNA for any specific gene that you'd like to knock down. And you can soak them in this media too, and the gene will be knocked out temporarily. So it's incredibly tractable. And because it's so fast to use, it's cheap, it's a great training tool for mentees, and also a really, really great tool for new investigators to generate massive amounts of data, mechanistic data that can translate to human work and potentially support grants for bigger projects as you're developing. So there's not enough I can say about how powerful this scientific tool is in this animal model. And we've done everything. So you can, of course, do molecular studies, but we've also supplemented the worms with ketones in recent experiments in the past 2 years to see exactly how ketone supplementation in the diet affects their function on the cellular and genetic level. We can track their neurons during this diet, and we're hoping that that translates to some work with our colleagues in human studies. And there is even a person who's developed an exercise protocol. So if you put the C. elegans in a media that's very viscous and they're swimming through it or moving through it, and it's essentially harder to move through because of the resistance of the material, you can use that as an exercise model. So yeah, I could go on for hours about the power of C. elegans.
It's fascinating.
It is indeed interesting. And we had an earlier guest on STEM Talk, Gordon Lithgow, episodes 129 and 130, who also uses C. elegans and spoke at length about their utility in his work.
So, Elaine, you've also done a lot of research into the mechanisms of osmosensing and adaptation in response to osmotic stress and infection. Can you give our listeners just a little bit of a short lesson on osmosensing?
So osmosensing is one very specific aspect of the response to osmotic stress or dehydration. I would say formally, we describe this to folks as the physiological response to perceiving a change in the environment. And so when you think about context, the most important kind of direct context where folks really study this heavily is in the physiology of fish. So there are fish that can go from freshwater to ocean environments and back and switch how they metabolize and how their cells respond to the different osmolalities on a very frequent schedule in their lifetime. And so osmosensing is the ability of the cells in that fish or organism to detect that switch in the environment and then elicit and cause changes at the cellular level that are appropriate to the environment that they're in at that time. So we've spent a lot of time trying to understand how C. elegans, and then, by translation to humans someday, how do the cells in C. elegans detect the actual change in osmolality to shift the gene expression from resting, healthy state, growth and reproduction to, okay, we've got a salt stress here. Now we need to mount a response to make osmolytes, and we need to make chaperones to protect any damage, and we need to change the expression of ion channels and so on. Like, where is the sensing mechanism and how does it work to switch on and off these genes? So that's osmosensing and what our goals are in that area of study.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research lab pioneering groundbreaking technologies aimed at leveraging and extending human cognition, perception, locomotion, and resilience.
As we've already discussed, your work in osmotic balance has sort of spanned the gap or spanned the range from basic research in worms such as C. elegans all the way through to elite level athletes. Could you characterize how well the work that you've done with C. elegans and other animal models, how well does that translate to human performance in human athletes or warfighters?
So at this stage, I think we're in that translational phase in the studies that we're pursuing. We have learned some really important things about the osmosensing and response in C. elegans that we are employing now in some of our studies. So I'll give you an example of that. So one of the things that we think is happening when cells stress dehydration, I'm going to focus there because that's where we've done the most mechanistic work, is that there is a metabolic change that leaves a gap cellularly that introduces through that gap a lot of opportunity for oxidative stress and damage, cellular damage through the buildup of reactive oxygen species. And so we know a lot of the genes that can mitigate that and control that in response to dehydration in C. elegans cells. And we have seen some of that same response in lymphocytes from human subjects who are dehydrated. And we have the RNA-seq data to confirm that it's happening similarly in both C. elegans and humans. The next stage, in terms of your question on how well it's translated, is what we're doing now. So one of the things that we're very actively pursuing now, and it's brought me back to my roots in nutrition in the past 2 years, is to take supplements or fruits, berries that are very high in antioxidant power, and to see if we can administer those to humans. And we are doing some mouse experiments also for pilot studies to understand whether consuming those prior to an exposure to dehydration stress or other stressors can mitigate the cellular response that we see after that stress. And so we think that this is just one component of a master mix that we would love to develop over the next 5 to 10 years that we could prescribe in a very specific way to induce a change in the cellular response. And so the translation is very real, and it's come back to a nutritional supplement. And I think there is opportunity to identify specific compounds and go towards a pharmacological route, but that part of it, we're certainly not there yet.
So, Elaine, you were part of a trial that looked at intracellular and surface heat shock protein 70 expression, and also looked at early apoptosis and also heat tolerance of lymphocytes during 11 days of whole body heat acclimation. So can you talk about how this study was conducted? Because it sounds like a really interesting study, and then also the significance of the findings from the study.
Yes, this study also was a really big learning moment in my career. So I did study in this whole kind of interest in cell resistance, stress resistance, and cytoprotection. heat shock proteins and the transcription factors that regulate their on and off switch in humans. And that was my dissertation work as a PhD student. It's something that I kind of kept with me through my postdocs. And this study my intention was for it to be this groundbreaking study that we could see this HSP70, which was at that time, I think, like a hot protein in the area of exercise science, and be able to say to people that as you become acclimated to heat every day over 11 days, and over that 11 days with the protocol that we used, you would also see accompanying physiological changes. So compared to day one, you would see, for example, that the sweat composition is more dilute, the sweat rate is greater, and you would see the rectal temperature the core temperature is much less for the same amount of exercise on day 11 versus day one. You'd see that the heart rate response is less on day 11 than it is day one. The subjects feel better, et cetera, et cetera. So there are lots of integrative physiological components that could confirm that that person is more resilient to heat stress on day 11 than day one. In the cells, I hypothesized with this study that you would also see this lasting expression and existence of HSP72, which is a protein that's really important for cytoprotection during cell damage. And it was named heat shock protein because it was discovered during heat shock experiments, this class of proteins. But we now know that HSP72 is important in cell protection against many different types of stress and cell insults. So what happened was we did not find that. And so every day the subjects would come in the morning, and in their blood samples, their HSP72 looked exactly the same. So I built in another separate kind of side study in that where, okay, maybe at baseline they're not, but are those cells that are circulating in the blood that we're interrogating, if we take them and put them in cell culture and give them another shock, another really dramatic heat stress in cell culture, does that show resilience and protection, at least with respect to HSP72? And we didn't find that. And so that was a moment where a very big name in HSP research, Larry Hightower, who's an emeritus faculty here and also a mentor of mine, basically counseled me through this journey with this paper and said, it's actually very informative that the answer is that it's not present. And he totally switched the way that I was looking at this story upside down and around. And we published in Cell Stress and Chaperones to say that you can have physiological adaptation without HSP72 being upregulated long-term. And everyone at the time that was really excited about HSP72 and said, this is going to be a pill that we could administer. This is something we can genetically manipulate. And all of a sudden, you're going to be Captain America. That's not the case. And that's been our story since. So it did change a little bit, but it's not the only part of the story. And that actually, now that I look back on it, makes sense with what we understand about cell stress responses and the way that we approach things with RNA-seq is that it's a huge landscape of genes that turn on and off during heat stress and dehydration. And even during acclimation, it's not that the cells stay in a stressed and kind of apprehensive state. It's that there's a dynamicism and a plasticity that is enhanced every time you're exposed to stress, and hopefully that makes you more resilient. So that paper was also a huge, and we tried to confirm it with 2 more papers that were published around that time to see if, we could replicate and see if there were other markers of heat stress protein function that were upregulated. We looked at transcription factors, clearance, expression on different types of cells, and we came to the same story that it's not only HSP72. And I still kind of fight that battle when we come to people who read the paper and have questions, or in review when we see other manuscripts come through or grants and things like that. So it's still an area that I think folks are coming to terms with understanding kind of more the broad scope and complex nature of this.
It's really interesting. So in 2012, you and Ken Strange followed up your award-winning paper in the FASEB journal with a paper in the American Journal of Physiology, Cell Physiology. And this paper demonstrated for the first time that inhibition of protein translation protects extant proteins from damage brought about by an environmental stressor. So can you talk a little bit about how this paper challenged some widely held views about chemical chaperones?
Yeah, I think this paper confirms what we had started to study in that lab that demonstrated that any stress cellularly reduces the rate of protein synthesis. And it's not just repair of damaged proteins that helps a cell or a person on a more global level respond and recover more quickly. It's also preventing the damage that occurs. So it's on both ends. And there's also complexity to both of those ends, but I'll just kind of leave it at that for this discussion. So we really challenged this idea that the emphasis during dehydration in cells is on damage repair and only on osmotic stress specific proteins, recipes that enhance the expression and function of things that protect against just the cellular osmotic stress and cell volume changes, that it's more global. And I'm surprised that it was, something that was reviewed pretty critically before we got it out there, because this data is out there in plant literature and other animal and organismal models for many, many decades. So there is lots of data, especially in plant research, where desiccation and dehydration and osmotic stress research has really gone so far in yeast work that that's true. So we really confirmed what's been out there in this model. And so that was really exciting for us. And it continues to drive kind of what we think about in our studies today.
Back in 2015, you worked with Jeff Volek and Stephen Finney and a number of other researchers on a paper that looked at the metabolic characteristics of keto-adapted ultra-endurance runners. And Jeff Volek is a friend of IHMC's and was our guest on STEM Talk episode number 43. Can you describe this study, its major conclusions, and specifically your role in the study?
Yes, so Jeff Volek was on my PhD committee and remains a really good mentor of mine. And of course, his work didn't necessarily overlap with mine when I was hired, but I was hired at UConn in 2012. And at that time, I was bringing to the Department of Kinesiology this knowledge and benchtop experience with RNA-seq. And so about 2013, just a year after I started my faculty position, Jeff came to me with this study, Jeff and Carl Marish, and they said, hey, we have this opportunity to study elite ultra-endurance runners who we will fly to UConn to do this challenge test. So it was a 3-hour treadmill run, and it's kind of remarkable when you think about the fact that most of these runners completed a marathon or more in that 3 hours, which was considered their 70% VO2 max, like not really their maximal effort. And so half of these subjects were on traditional high carbohydrate diets, the diets that you hear folks always tell your high school runners, you got to carb load before your race. And then the other 10 were consuming for at least 3 to 6 months, and much more in many cases, really low carb ketogenic diets and still performing at the elite level. So I think that fact alone is really compelling, right? So we're bringing in elite athletes who are world record holders, and they're already consuming this non-traditional, very controversial diet when it comes to the world of endurance training. So our job, I think, at that point was to understand how they're able to achieve that. And cellularly, I think my role was to suggest, at that time we were working with a company called Ocean Ridge Biosciences, which is in Florida. And that was where I collaborated to do a lot of our RNA-seq sequencing and analysis, because at that time they had one of the fastest turnarounds for data analysis on a very reasonable price scale, which is always important. And I had already completed about 6 RNA-seq experiments with this company. So I said to Jeff, if you're going to get all this tissue, they were getting biological sample of all different sorts of sweat, saliva, blood, cells from blood, adipose tissue, muscle tissue, stool samples for microbiome work, and so on. And I asked if we could take some of that skeletal muscle and do RNA-seq experiments on it to see if, on a global level, the athletes who are consuming low carbohydrate ketogenic diets have a different pattern of baseline life in their skeletal cells, and what happens during exercise and after exercise and then during recovery. Is it a totally different experience for those skeletal muscle cells because of what the host, the athletes, are consuming long term? And so that it was, at that time, we had a hugely captive population, right, to be able to have access to these athletes. And then tissue, which is incredibly invasive, in a really well-designed controlled study in the lab. And then, of course, the technology that allowed us to capture this data, which at that time was, we would get back up on Blu-ray discs, and there are 64 Blu-ray discs worth of data. So to these days, obviously, you store those on servers, but that was really cutting edge and huge. And we're still, many years later, kind of finalizing that paper.
Hmm. It certainly was an interesting study, and I remember when it took place. I knew several participants in the study, and they described it as, in some regards, as unique and painful, actually, a few times.
Yeah.
Last year, you were one of the lead authors of a study that Jeff also participated in that addressed how endocrine response to strenuous experience in the heat can serve as a sort of indicator of the amount of strain that the person is experiencing. Could you talk about the findings and the significance of this study?
Yeah, I think this study identified a mechanism that I haven't been able to kind of chat about today yet, that's more high level upstream. And it really is supported by all the genetic and genomic changes that might happen cellularly and integratively with the nervous, endocrine, and immune system during stress. But this study really focused on that big picture endocrine response during dehydration in humans. And I think that's really important because that's something that we can't get from the animal models and the cell culture. And it's a piece of the story that I think contributes a uniqueness to human stress. So the main finding was that we were able to see endocrine changes that can be attributed to the stress condition and rehydration, and gives us tools to basically have biomarkers or bigger markers of that stress response in the blood to track it. And hopefully, if we start to administer protocols for hydration, timing, volumes, composition of the fluid, and whether it's oral or IV and so on, all these variables that are pretty fluid and, not standard to people right now, that we have blood biomarkers or tools that can validate that they're working. And so that study really provided that.
So, Elaine, I think we're going to transition to discussing your lab at UConn, which is called the E.C. Lee Laboratory. And it says on your website that you and your colleagues are passionate about studying the physiology and genetics of resilience. Can you talk about how your lab uses genomic technologies to ask questions about what makes elite athletes elite?
Yes. So my lab includes individuals who are incredibly resilient in their own different worlds. And everyone is really motivated and self driven about this topic. But we all have different perspectives that give us some practical application. We've had many, many military veterans in the lab, which I'm really excited about. And so, essentially, in any of the different contexts for the different studies that we've pursued to answer questions about resilience to stress and ways to improve recovery, speed it up, the nature of it. We also, every time, collect immense amounts of biological sample to understand how the more resilient animals or individuals are different, at least in the genetic profiles, the protein profiles, the biomarkers in blood, and then how that relates to the physiological and functional ways that we see that resilience manifested. So the genetic piece is another thing I think that ties together every single study that seems so diverse. We have used a lot of RNA-seq, and now we're going into more single-cell genomics technologies to interrogate specific cell types. And a lot of times we have focused on cells in circulation. So the lymphocytes, the peripheral blood mononuclear cells, and neutrophils that circulate and survey and basically assist with immune defense. And so there's always that piece of immune system resilience in there because, practically speaking, this is a really easily accessible tissue to us. And so if we're lucky enough, we will certainly look at more invasive samples like skeletal muscle, as we did with Jeff Folick. But many of the times we're doing RNA-seq experiments in circulating cells to give us an indication of where to go when we have more invasive samples. And so we've done everything from heat stress isolated to heat and dehydration, dehydration alone, combination stress recovery protocols, acclimation, the influence of gear. So what you're wearing, for example, like football uniforms, down to your sneakers. And so we're always integrating all different aspects of what that athlete or military warfighter experiences on the genetic mechanisms.
That's awesome. So it might be good here to talk about some of your lab specific projects. So I wanted to start with one project that's looking at aronia fruit and stress resilience and longevity. Can you talk a little bit about that?
Yeah, so this branch of our laboratory, we have two projects that are in nutrition, and it really has been kind of an evolution of my lab to finally make a transition from all this characterization and mechanistic work to understand what we can do that's not a drug, hopefully, and a little bit less invasive and something that people can employ easily to enhance resilience and improve recovery day to day. And so we focused, because of our collaborations, on ketone supplementation, and then more recently, because of the mechanisms that we were seeing in response to cellular stress, antioxidants. And this collaboration and this focus on aronia came about, again, really quite serendipitously and not intentionally, because in one of my offices in a wet bench lab that we do a lot of genomics work, my next door neighbor happens to be a plant scientist, Dr. Mark Brand, who grows aronia fruit in the UConn research laboratory fields here, the farms. And one of his areas of work is to genetically modify aronia fruit. And at that time, a PhD student was trying to work on making aronia berries bigger and sweeter by crossing them with a pear. And so if you're familiar at all with aronia, it's, first of all, the name is terrible. It's called a chokeberry. So we just call it aronia because that will never do to, kind of market this. But the black chokeberry is incredibly high in antioxidants like anthocyanins, proanthocyanins, and polyphenols, more so than blueberries, blackberries, tart cherries, and so on. But what has made it challenging for this berry to gain some traction in the supplementation world is that it's incredibly tart. It's very bitter, and that comes from, some of the composition of the berries in the skin. So Mark Brand was able to give us powder extract that we could use in C. elegans, and we were finding that on a dose response to different prescription regimes, that the worms consuming aronia and living on aronia were incredibly resilient to toxic stress with chemicals, dehydration stress, heat stress, and so on. And so we've been tightening up that data recently, and we included it in a grant that we recently had funded by DoD to see if we could administer aronia berry powder or drink to enhance response to heat acclimation among military warfighters prior to their going to deployment to hot and humid climates. So that's the journey with that fruit. And what we've also found is that we have a very famous expert here at UConn in the area of antioxidant fruits, Dr. Ok Chun in nutritional sciences. And she also studies similar things in clinical contexts with black currant. So now we've kind of been doing side-by-side studies of aronia, black currant, and other berries to see what we can recommend. And so that's also being included. So that's kind of the journey with that project and that supplement, and we're still kind of refining the form, the extract process, the berry species that we use, the seasons and the farms that they come from, and seeing if we can identify ones that really show marked genetic changes in response to stress that show that the worms and our humans are tougher and more resilient and recover faster.
In the case of humans, performing in the heat and the humidity, but presumably mostly heat, what have you found?
We have just started this study. So this study launched last week. And so we don't have any results from the human studies, but we're doing RNA-seq experiments in the skeletal muscle and the circulating lymphocytes. And our targets are based on the C. elegans work. So we anticipate that we'll see differences in the supplemented individuals in pathways that show resistance to heat and dehydration stress and oxidative stress in the C. elegans. Many of them are related to general protein synthesis like ribosomal function, housekeeping genes, and then, of course, damage and repair proteins.
That's really interesting.
Yeah, absolutely. So, Elaine, you have another project focused on enhancing warfighter resilience, which is something that is studied extensively here at IHMC as well. Can you tell us more about your project?
Yes. So this is one of two that we are pursuing. And one aspect of this project that is especially important as we're moving forward is the study of resilience differently in men and women, biological sex-wise. So we think that some of the mechanisms that we've studied have been limited to studying men, or obviously in C. elegans with the hermaphrodite animal model, there's not much opportunity to understand the integration with women versus men in stress response. So Those two studies we will pursue have a sub-aim of studying how men and women respond differently. And then what we are doing is to introduce repeated extreme physical and environmental stressors, and heat is one way that we can do that in a really controlled way in the laboratory. And so we are testing the outcome functionally. So it's really truly directly applicable to things that the warfighter will experience. So I'll give you one example of a test that we intend to employ. If you're acclimated versus unacclimated, during your first exposure to heat stress and exercise stress, in an unacclimated individual, what is your experience in terms of cognitive function? So when you perform on stressful cognitive tests like reaction time, memory, and then visual vigilance, like paying attention for 40 minutes at a time for a very small cues, like a pinpoint cursor, a white cursor showing up on a black screen, and then other more complex and integrated tasks like a simulated plane flying exercise or a rifle marksmanship exercise that allows us to capture the success and failure rates of your performance using computer software. And then physiologically, so what does your sweat composition look like? How quickly do you start to sweat? What's the rate of sweating? How fast does your core temperature rise? How quickly does your heart rate rise? How well can you maintain a rested state under these conditions? We anticipate that an acclimated individual who's been exposed to at least 5 exercise heat exposures will display better functional and cognitive performance. And then in the tissue, we're going to try and understand if any of those performance differences are predicted or related to the genetic changes that we see. So we aim to do this over the next 4 years. We have one 3-year project and another one starting next year that will allow us to really understand on so many levels what enhances resilience during this repeated stress exposure, and then what things might be behind that, at least in skeletal muscle, adipose tissue, and the immune system, and then, of course, in plasma circulation, the endocrine system, and systemic signals.
So, Elaine, you're also studying the effects of exercise and supplementation interventions on immune function to better understand the mechanisms by which exercise can enhance immune function. So, what can you tell us about this project and this research?
So initially, I think in my career, I started focusing a lot on immune function because I was very interested in the immune system. But I think the immune system and function and resilience to infection is a really good readout of whether you are resilient and healthy at a certain point, given everything else that an athlete is experiencing at that point. I always tell kind of new trainees or my students or people coming to the lab when I'm trying to describe what we're studying, that how many of you have experienced a situation in which you do well, you're overcoming many, many obstacles and challenges, psychological, physical, physiological, emotional, etc. And you do okay, but once a project's done, you get sick. And, so many of my students in class raise their hand and say, yes, I get sick after finals every semester for about a week, and it's terrible. And I think the immune system is a very expensive system energetically, and it's one that's probably the best readout in terms of how healthy you are at a given state. How much have you recovered from stress, or how much are you basically cellularly just triaging and just managing? And at some point that will fail. So we use that system, especially the circulating surveillance system and the reactivity of those cells to subsequent infection and culture, to understand whether an athlete who feels okay on all other fronts or a military warfighter who's able to do well is really suffering some way cellularly, subcellularly, that we can detect using the immune cells. And so that's kind of where we're going with this project and exercise and many of our projects, because we think that, especially among our heroes, right, our elite athletes and our warfighters, they do fine on reaction time tests. They do great on visual vigilance tests. They can overcome a lot of fatigue and everything else that they feel physically and sometimes even physiologically to do the job. But what if that person who's just managing to that level is exposed to coronavirus, right? COVID-19. How well does that person respond to that infection might be a real subtle circumstantial signal of whether that person is really recovered and responding well to stress after stress after stress. And I think for our athletes, that's the ability to distinguish really effective overreaching and training adaptations versus overtraining.
So, Elaine, you're also doing research to better understand how transfer ribonucleic acid, or tRNA, and aminoacyl tRNA synthetases relate to the switches that regulate the genes that are important in protecting cells during stress and aging. And so, we were wondering if you could talk a little bit about this project and also give our listeners an overview of how tRNA are responsible for cell survival during stress.
Yes, so this tRNA aspect of our research has become part of the collection of outcome variables that we measure because in the 2012 and on work, 2010 to 2012 work with Kevin Strange, when we saw that C. elegans were stressed osmotically and dehydrated, among the things that controlled the rate of protein synthesis and shut it down a little bit, or reduced it, I should say, to allow stress response genes to be expressed and the emphasis to be placed on responding to the stress at hand. We also observed that tRNAs were really important in that process. So we started this idea that it's not just about controlling the proteins that translate protein, ribosomes and eukaryotic initiation factors, for example, but also the ingredients that make up protein are somehow involved in this process of controlling what the emphasis is on during stress. And so we did some experiments that were really exciting at that time and new, where we knocked down or knocked out tRNA synthetases that bring tRNAs to the amino acid that they match up with, the cognate amino acid. And then we also selectively deprived worms of certain tRNAs in their diet. And we were finding, really surprisingly, that even though the worms were experiencing lower metabolism and reduced protein synthesis rates, which you would think is terrible for them, and it was in some ways, they were living longer and they were very resilient to stress. And so, what we now understand and what we include in some of our outcome measures and biomarker work is tRNA processing enzymes and tRNAs themselves, to understand how these small molecules are playing a role in stress sensing and resilience to stress and switching on and off genes when they should be switched on and off. And our working hypothesis now is that the concentration of tRNAs and where they are in the cell is an important signal for the cell to know whether it's stressed or not, and then to change what's being expressed, what proteins are being made, and to put that cell in like an emergency stress state versus a resting state and deal appropriately with what's at hand. And this is also matching with a lot of the data that show that amino acids and tRNAs are not just about building blocks for proteins. They go to the brain, they bind receptors, they control lots of other functions, they signal immune activation. And so we're really excited to jump in this area. And there are also, again, quite serendipitously, about 2010, there were new development in tRNA microarrays and sequencing that allow us to understand what tRNAs are present, what's their sequences, how many of them there are, what is the concentration, and so on. And now we're interrogating that in direct diet studies with aronia fruit and blackcurrant. And then, of course, in the world of ketones, this is so huge and yet unstudied. So we're really excited to pursue that outcome variable in some of the studies we're looking at right now.
As we have noted, you've published several papers in the various omics fields. And this topic has long held promise, but it's really yet to deliver on many actionable points to improve human performance. What do you see in a nutshell? I mean, just briefly, what do you see as what needs to be done now to close that gap?
I think that we're doing that right now in 2021. So if you think about the advent of high throughput sequencing and all the equipment and data science that comes with that, we're really only about 12 years in, I think. And I think a lot of the work in this past 10 to 12 years has been on characterization, understanding the diversity and expression diversity and contextual diversity of these omics. And now what you see in 2020 and 2021 are all these fields, master's degrees, PhDs in data science and machine learning and algorithms, and also in technology that allows us to integrate omics, so not just transcriptome, but epigenome. And there's so many omes now that it's kind of mind-boggling how many there's an envirome and, all these different omes. And understanding how those integrate so that we can target them. So I think we're at the threshold right now, this past couple of years, in a movement towards that application and to manipulate what we have been trying to understand for the past 10 to 12 years. That's going to require a huge emphasis on collaboration among benchtop applied scientists and clinical scientists, and then mathematicians, algorithm specialists, and data scientists. And I think that is occurring successfully in some models, and we certainly are trying to do that here.
A growing body of your work could be characterized as network physiology. Can you briefly tell us what that is and elaborate just a little bit on that?
Yes, so network physiology is not new, and translational omics is another kind of catchphrase that we now use to kind of describe the evolution of what we've been doing. Briefly, it is the integration of multiple omics outcomes with physiology and function to try and understand networks of how things come together in an individual. And so there have been two journals, Frontiers in Physiology and Physiological Genomics, that I've participated in starting new sections for papers in these areas. And then many scientists, more senior and also junior than me, that are joining this field where we do what I just described previously, which is integrate all of the omics work with the functional and physiological outcomes to understand customization and individualization of response, healthy state, recovery, and then in the future, I think in the next 5 to 10 years, how can we enhance that for people individually? And then what are ways that work for big groups of people? So that's essentially, it's integrative, and I think it's certainly not new, but it is supported by the advent of a lot of new technology and data science abilities.
Electrolytes and electrolyte replacement have been a hot topic in sports performance now for some years, and they seem of increasing popularity and interest. Where are we in our ability to personalize electrolyte replacement, and how does the baseline diet and individual differences in electrolyte loss factor into this? Are we able to leverage this knowledge for better performance? Where does this all stand now?
So we're at the frontier, and we are able to do quite a lot now, and that's actually one of the big parts of our big 2 DoD studies that we're pursuing right now. And I would say that that builds on the last question about network physiology, because not only are we integrating multiple fields of research, but we're now also collaborating, at least in our field, with engineers who can develop tools to monitor in real time and validate to biomarkers and what's happening in the tissue. And so in the electrolyte space, we've been collaborating recently with Epicor, which is the company that has developed the Gatorade electrolyte patches that you may have seen. They're a really simple readout. It's a patch that's disposable. You put it on the skin, and it's a red, yellow, green system for danger to healthy electrolyte concentration in the sweat. And that's a really good readout of what's happening physiologically to result in whatever outcome occurs in your sweat. So when it comes to consumption that enhances that balance to support performance, the impact of diet, the impact of your microbiome uniquely in relation to all aspects of your health, before an exercise, after an exercise, with training, and how that affects your fluid electrolyte balance is something that we can now study not only in the blood, but also in sweat. So with Epicor, they've developed a new system that allows sweat collection in a really clean and safe and uncontaminated way. And I actually have a student, Kyle Mahoney, who's doing his master's thesis on some really cutting edge sweat characterization. So we're doing proteomics analysis, microvesicle, isolating what's in the microvesicle is one of our big goals, and seeing how complex sweat is to be able to tell us about whether some of the interventions that we're administering are working or not to improve fluid electrolyte balance during exercise and recovery. So we're there, and there are certainly many other groups doing it. And of course, Gatorade Sports Science Institute, GSSI, is at the cutting edge of that because they have a very direct corporate industrial aim related to that. And so we have so many different folks in academia, industry, and the corporate world working to answer these questions.
So, Elaine, when people ask you what they can do in terms of exercise, diet, and supplementation to improve either their cognitive or their physical performance and even their health span, what do you advise?
So that's a great question, and I get that all the time, especially from media outlets too, who kind of come to me for this. I always give the disclaimer that there is no one answer, and I really believe that that's going to be something that's confirmed more and more as we get more data. And so there's no magic bullet, and I think anything that suggests there is maybe missing a big population or in some ways missing some aspects of cellular stress response. What I do advise athletes, coaches, and folks who come to me is that monitoring your own individual performance in whatever way that looks like for you that's relevant to you. And essentially, it's a movement in citizen science, right? It's to take your own data and track it over time systematically. So I'll give you an example. Erroneous supplementation may be very powerful, and we can demonstrate it in C. elegans. But the trick to identifying whether it works in humans to give a very specific dose and say, oh, you need to drink, 300 mils in the morning every day is pretty limited in terms of the application and really what the data mean. And so for individuals who are experimenting in their own diets with supplements or changing their diet to low carbohydrate ketogenic diet, for example, is to consult with an expert, first of all. I think there are companies like those started by Jeff Follick that offer that. And then monitor your own data and see what does have an impact and what doesn't. And basically, over time, do dose monitoring, prescription changes. And I think that's where the customization is going to become really effective from both the researcher perspective and the citizen science perspective. And then just kind of anecdotally into the lay population, I always communicate to athletes and coaches that they should trust and listen to their own bodies. So it seems like a very soft science thing to say, but the truth is, even if we demonstrate something cellularly and genomically, there's so much complexity that we don't understand about a person's everyday life that he or she is experiencing that has to be, included in that equation, that consideration. And so that's my advice, is to track your own data, pay attention to your baselines and where that deviates, and then use the tools that you think are going to help you in a very strategic way as you monitor those things. And that might come with some need to train people on how to monitor and use wearables and all these new tools that are coming out effectively.
So, Elaine, we're going to ask you now, kind of stepping away from the science, what do you do in your spare time? And we understand that you have 2 toddlers, and just kind of guessing that keeps you quite busy.
Yes. So I have a 5-year-old and a 3-year-old, and they are most of my spare time. But I have found ways to do the things that I love to do with them. So I play classical piano and violin, and I've done so since I was about 4 years old. And I am now using that hobby to basically teach my kids. So in my free time with them, my son started cello and my young daughters started violin, and I have my own violin, and we play music together. I certainly love to run and exercise, so training is a really big part of my life. I do train for events, most of the time not necessarily competitively, but for the experience. So one of my goals over my life is to do some of the most scenic marathons that Runner's World, that magazine kind of reports. So the next one on the list is in Missoula, Montana, and we're hoping to do that either next summer or the summer after. And so, yes, music, art, and right now my kids are my hobbies.
Yeah, that's fantastic. And so you obviously love what you do, and your lab has a staff of around 24 people right now, from what we understand. And I understand that you love your lab and job so much that you wake up excited every day to do good science, which you can kind of tell just from talking to you today.
Yeah, I feel so lucky, and I tried to tell that to my students, especially my pre-med trainees, who you can kind of sense that they don't really want to do it and they're getting external pressure to pursue that. There's so much expectation, I think, to do certain things in a certain way. But everything that I've experienced in my scientific journey, even if it hasn't been intended, I've been able to see the value of it and add some perspective to what I'm doing. And so I feel so lucky. There's no aspect of my day-to-day job in science that I don't love. I love to write. I love to come up with ideas. I really love organizing and putting together grants. I think the most exciting thing recently that I've been able to do is to collaborate with folks like those at IHMC and other institutes and, just talk science. And that really is so, so fun and exciting. And also, I think, innovative. So I feel very fortunate that I get to do what I love every day. And sure, there are some stressful things, but I think probably I'm one of very few people who just, at night I can't go to sleep because I'm excited about what I'm going to do the next day, and I have complete control over the direction of my research, and it's just really fun, and I get to work with some incredible trainees at all levels.
That's so fantastic, and you're certainly doing amazing work and making such an impact through that research. We're really looking forward to following your lab's research in years to come. You guys are doing some really exciting work, so thank you so much for joining us today, Elaine.
Thank you for having me. It's been really wonderful to talk, and I'm just so excited to be part of IHMC STEM Talk.
Thank you, Elaine. It was great.
STEM Talk. STEM Talk. STEM Talk. STEM Talk.
So that was a lot of fun, Ken. I have to say, Elaine's enthusiasm for science and for her lab is absolutely infectious. And I also have to say that I'm a Marvel Comics fan myself, and I love how Elaine is using science to figure out ways to help our real-life superheroes, and that's how she put it in the interview, in the military improve their resilience and performance. It's such fascinating work.
It is done, and some of the work she's doing is quite similar to some of the research we do here at IHMC. So it's fun to talk with her, and it's going to be really interesting to follow Elaine in her lab over the years as it develops. We're looking forward to keeping an eye on it.
Yeah, I definitely agree with you, Ken, on that one. So if you enjoyed this interview as much as Ken and I did, we invite you to visit the STEM Talk webpage, where you can find the show notes for this and other episodes at stemtalk.us. This is Don Carnegie signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go